Assalamu alaikum. In this series, we will discuss about the most repeated genetics engineering MCQs. So let's begin. MCQ number 23. Genetically engineered humulin is produced by A. Salmonella, B. Bacillus thuringiensis, C. Escherichia coli, D. Agrobacterium tumefaciens. The correct option is C. E. coli. MCQ number 24. Interferon produced by a virus infected cell is A. Lipid, B. Amino acid, C. Polysaccharide, D. Protein. The correct option is D. Protein. MCQ number 25. Monoclonal antibodies are produced by A. PCR technology, B. Hybridoma technology, C. DNA fingerprinting, D. Radioactive isotopes. The correct option is B. Hybridoma technology. MCQ number 26. Hybridoma technology was developed by A. Keremolis, B. Singer and Nicholson, C. Kohler and Milston, D. Nathan Smith. The correct option is C. Kohler and Milston. MCQ number 27. According to Human Genome Project, human genome contains approximately A. 30,000, B. 50,000, C. 70,000, D. 1 lakh. The correct option is D. 1 lakh genes. MCQ number 28. The process of enzymatic degradation of xenobiotics is known as A. Bioelastic, B. Bioremediation, C. Enzyme operation, D. None of these. The correct option is B. Bioremediation. MCQ number 29. The artificially manufactured chemical pollutants are known as A. Pseudobiotics, B. Antibiotics, C. Xenobiotics, D. Chemobiotics. So the correct option is C. Xenobiotics. MCQ number 30. The bacterium genetically engineered to enhance the commercial production of alcohol is A. Bacillus, B. Pseudomonas, C. E. coli, D. Zymomonas. So the correct option is D. Zymomonas. MCQ number 31. The preservation at a very low temperature is known as A. Cryopreservation, B. Xenopreservation, C. Thermopreservation, D. Pseudopreservation. So the correct option is A. Cryopreservation. MCQ number 32. In a cryopreservation, the temperature is maintained at A minus 96 degrees centigrade, B 100 minus 196 degrees centigrade, C minus 246 degrees centigrade, D minus 273 degrees centigrade. So the correct option is B minus 196 degree centigrade. MCQ number 33. In 1996, Ian Wilmot and his colleagues first produced A. golden rice, B. Basta wheat, C. Dolly sheep, D. Rosy cow. So the correct option is C. Dolly sheep. MCQ number 34. The cloning of Dolly surrogate mother is the A. first fertile female, B. second fertile female, C. third fertile female, D. fourth fertile female. The correct option is C. third fertile MCQ number 35. Which cells of the female donor sheep were taken in cloning of Dolly? A. Egg cell, B. embryo cell, C. tail cell, D. udder cell. The correct option is D. udder cell. MCQ number 36. The genetic ability of a cell to regenerate into a whole plant is called A. Transformation, B. Totipotency, C. Transfection, D. Genopotency. The correct option is B. Totipotency. MCQ number 37. The phenomena of production of haploid plant from pollen is known as A. Androgenesis, B. Gynogenesis, C. Embryogenesis, D. Microgametogenesis. The correct option is A. Androgenesis. MCQ number 38. The temperature of a culture room is generally maintained at A. 20 plus 2 degrees centigrade, B. 22 plus minus 2 degrees centigrade, C. 25 plus minus 2 degrees centigrade, D. 27 plus minus 2 degrees centigrade. The correct option is C. 25 plus minus 2 degrees centigrade. MCQ number 39. In tissue culture, autoclaving is done at a pressure of A. 5 psi, B. 10 psi, C. 15 psi, 20 psi. So the correct option is C. 15 psi. MCQ number 40. HEPA, high efficiency particulate air filter, can filter particles larger than A. 3.0 micrometer, B. 0.3 micrometer, C. 0.03 micrometer, D. 0.004 micrometer. Correct option is D. 0.004 micrometer. MCQ number 41. The surfaces of the equipment associated with tissue culture are sterilized with A. 70% alcohol, B. 80% alcohol, C. 90% alcohol, D. 100% alcohol. The correct option is A. 70% alcohol. MCQ number 42. Which of the following is not a surface sterilizing agent for tissue? A. Calcium hypochlorite, B. Sodium hypochlorite, C. Mercuric chloride, D. Magnesium chloride. The correct option is D. Magnesium chloride. MCQ number 43. For production of shoot in the calysaxine cytokinin should be A. 1, B. less than 1, C. more than 1, D. axine of cytokinin not required. The correct option is B. less than 1. Number 44. In tissue culture, the carbon source of culture media is generally A. starch, B. inulin, C. sucrose, D. proline. The correct option is C. sucrose. MCQ number 45. Agar is obtained from Gelidium manso, which is A. red alga, B. brown alga, C. green alga, D. blue-green alga. The correct option is A. red alga. MCQ number 46. The pH of culture medium is generally maintained at A. 4 to 5, B. 5 to 6, C. 6 to 7, 7 to 8. Correct option is B. 5 to 6. MCQ number 47. The first somatic embryogenesis was produced in A. radish, B. sweet potato, C. rye, D. carrot. The correct option is D. carrot. MCQ number 48. For somatic embryogenesis, generally the growth regulator used is A. IIA. B. IBA. C. 2,4-D. D. 2,4,5-D. So the correct option is C. 2,4-D. CQ number 49. Indirect androgenesis involves formation of A. Callus. B. Proembryo. C. Ovule. D. None of these. The correct option is A. Callus. CQ number 50. Gynogenesis was first reported in 1976 in A. Brinjal. B. Tomato. C. Wheat. D. Barley. The correct option is D. Barley. MCQ number 55. Somatic hybridization refers to A. Ovule fusion. B. Pollen fusion. C. Protoplast fusion. D. Fusion of pollen of one species and egg of another species. The correct option is C. Protoplast fusion. MCQ number 53. Pomato is produced by protoplast fusion between A. Solanum tuberosum and Lycopersicum persivianum. B. Solanum melangina and Lycopersicum esculentum. C. Solanum melangina and Lycopersicum perquianum. D. Solanum tuberosum and Lycopersicum esculentum. Correct option is D. Solanum tuberosum and Lycopersicum esculentum. CQ number 54. Crown gall in plants is induced by A. Agrobacterium tumefaciens. B. Agrobacterium rhizogenes. C. Bacillus thuringiensis. D. Phytophthora infestans. The correct option is A. Agrobacterium tumefaciens. MCQ number 55. Artificial seeds are produced by encapsulating somatic embryos in beds of gel containing A. Calcium pictate. B. Calcium alginate. C. Calcium phosphate. D. Calcium chloride. MCQ number 56. Raphanobbrassica is produced from a cross between somatic cells of A. Potato and radish. B. Radish and carrot. C. Radish and cabbage. D. Carrot and cabbage. The correct MCQ number 57. Triticale is a hybrid between A. Wheat and rice. B. Wheat and maize. C. Maize and rye. D. Wheat and rye. The correct option is D. Wheat and rye. CQ number 58. When a plant contains foreign gene and genes in addition to its normal genome, then it is known as A. Clone. B. Transgenic. C. Hybrid. C. Hybrids. So the correct option is B. Transgenic. MCQ number 59. The gene which is transferred from one organism to another is called A. Transgene. B. Regulatory gene. C. Constitutive. C. Pseudogene. The correct option is A. Transgene. CQ number 60. The first transgenic plant introduced to market was A. Golden rice. B. BT cotton. C. Flavor saver tomato. D. Higher protein content potato. The correct option is C. Flavor saver tomato. Number 62. Name the transgenic plant whose leaf can act as a vaccine against hepatitis B. A. Potato. B. Tomato. C. Datura. D. Tobacco. The correct option is D. Tobacco. 63. Plant vaccine against HIV-1 has been developed through cowpea mosaic virus. A. Tobacco. B. Barley. C. Rye. D. Carnation. The correct option is A. Tobacco. MCQ number 64. The weeds which carry transgene from herbicide tolerance are called A. Transweeds. B. Herbiweeds. C. Superweeds. D. Xenoweeds. The correct option is C. Superweeds. MCQ number 65. The genetic material in retrovirus is A. DNA. B. RNA. C. Protein. D. cDNA. Correct option is B. RNA. 66. The first transgenic animal reported was A. Sheep. B. Cow. C. Rabbit. D. Mouse. Correct option is D. Mouse. MCQ number 67. The name of the transgenic cow which carry human gene for alpha lactalbumin in production of human protein rich milk is A. Rosie. B. Dolly. C. Lily. D. Ginny. Correct option is A. Rosie. MCQ number 68. The gene for antifreeze has been transferred from A. Sheep. B. Rabbit. C. Fish. D. Spider. Correct option is C. Fish. 69. The name of transgenic animal that can produce silk strands along with the milk. A. Cow. B. Buffalo. C. Goat. D. Sheep. The correct option is C. MCQ number 70. Interferon, human growth hormone, fertility hormone, etc. are produced by transgenic microbe. A. Agrobacterium. B. E. coli. C. Bacillus. D. Pseudomonas. The correct option is B. E. coli. MCQ number 71. The transgenic microbe which carry NEF gene of Klebsiella. A. Pseudomonas fluorescens. B. Pseudomonas patida. C. Bacillus thuringiensis. D. E. coli. The correct option is D. E. coli. '72. Clones can be produced A. In vitro only. B. In vivo only. C. Both in vivo and in vitro. D. Neither in vitro nor in vivo. The correct option is C. Both in vivo and in vitro. MCQ number 73. Magic bullets are A. Monoclonal antibodies. B. Polyclonal antibodies. C. Monoclonal antigens. D. Polyclonal antigens. The correct option is A. Monoclonal antibodies. MCQ number 74. Bacterial resistance to antibiotics is a genetic trait carried in bacterial. A. Genome. B. Episome. C. Centromere. D. Plasmid. The correct option is D. Plasmid. 75. Hybridization of messenger RNA with DNA can reveal the segments of and trans or exons of a DNA molecule. When the magnification is high enough to see the molecules, the introns appear as A. Separate segments. B. Triple-strand nucleic acid molecules. C. Single-stranded loops in a double-strand molecule. D. Bar bodies. Correct option is C. Single-stranded loops in a double-stranded molecule. MCQ number 76. Each restriction endonuclease cleaves a molecule only at A. the ends of genes. B. a particular nucleotide sequence. C. methyl group. D. centromere. The correct option is B. a particular nucleotide sequence. 77. The formation of phosphodiester bond between deoxyribose of one DNA fragment and the phosphate of another DNA fragment is facilitated by A. DNA ligase, B. Endonuclease, C. Fermiase, D. DNA phosphodiesterase. The correct option is A. DNA ligase. 78. A eukaryotic chromosome differs from a bacterial chromosome in having A. Start and stop signals, B. Thymine instead of uracil, C. Reverse transcriptase, D. Intrans. The correct option is D. Intrans. 79. A fragment of DNA cut by restriction in the nucleus forms bonds with other DNA molecules that have A. Been fragmented by the same restriction into nucleus, B. Sticky ends, C. Blunt ends, D. Cohesive ends. Correct option is A. Been fragmented by the same restriction into nucleus. 80. The sticky ends of a cut DNA molecule contains A. Methyl group, B. Calcium salt, C. Unpaired bases, D. Ribose sugar. The correct option is C. Unpaired bases. MCQ number 81. Restriction endonucleases are synthesized by A. Yeast only, B. Bacteria only, C. Both yeast and bacteria, D. Eukaryotic cell only. The correct option is B. Bacteria only. 82. A bacterium adds methyl group to its DNA in order to A. Turn its gene on, B. Transcribe many genes, C. Clone its DNA, D. Protect its DNA from its own restriction enzyme. The correct option is D. Protect its DNA from its own restriction enzyme. 83. An advantage of using yeast rather than bacteria as recipient cells for recombination of eukaryotic DNA is that yeast can A. Remove methyl group, B. Reproduce more rapidly, C. Excise and trans from the RNA transcript, D. Produce restriction enzymes. The correct option is C. Excise and trans from the RNA transcript. 84. A restriction in the nucleus cleaves bonds between A. Base pairs of a DNA molecule, B. Introns and exons of a DNA molecule, C. Sugar and phosphate components of a nucleic acid molecule, D. Base pair of a DNA RNA hybrid molecule. The correct option is C. Sugar and phosphate components of a nucleic acid molecule. 85. In situ hybridization is used to A. Locate a gene on a chromosome, B. Clone a gene, C. Remove plasmids from a cell, D. Remove mutations from a chromosome. The correct option is A. Locate a gene on a chromosome. MCQ number 86. The technique of recombinant DNA first became available in the A. 1950s, B. 1970s, C. 1980s, D. 1990s. Correct option is B. 1970s. 87. In DNA hybridization, the hybrid is formed of A. One double strand DNA and one single strand RNA molecule, B. Two double stranded DNA molecules, C. Two single stranded DNA molecules, D. One single stranded DNA and one single strand RNA molecule. Correct option is C. Two single stranded DNA molecule. 88. A piece of DNA molecule used to find a gene by forming a hybrid with it. It is called a vector, B. Restriction sequence, C. Clone, D. Probe. Correct option is B. Restriction sequence. CQ number 89. A gene carried by recombinant DNA is cloned when A. It is transcribed, B. Its host bacterium divides by binary fission, C. It is hybridized, D. It is fragmented by restriction enzymes. 90. A laboratory technique for separating molecules on the basis of size and electric charge by measuring their rate of movement through an electric field is known as electrophoresis. D. Chromatography. C. PCR technique. D. Ultracentrifugation. Correct option is A. Electrophoresis. 91. Complementary DNA, cDNA, is produced from A. Single strand DNA, B. Double strand of DNA, C. Messenger RNA, D. Transfer RNA. The correct option is messenger RNA. 92. The enzyme that facilitates the synthesis of cDNA is A. Transcriptase, B. Reverse transcriptase, C. DNA ligase, D. DNA polymerase. The correct option is B. Reverse transcriptase. Number 93. The position of a probe on a chromosome is easily detected by making the probe with A. Phosphate, B. Deoxyribose sugar, C. Heavy water, D. Radioactive atoms. Correct option is D. Radioactive atoms. 94. A strain of bacteria is known to synthesize protein X, by which experiment one can isolate the gene that codes for this protein. A. Shotgun experiment, B. Transformation experiment, C. cDNA experiment, D. Electroporation experiment. 95. The number of restriction endonuclease known as approximately A. 100, B. 300, C. 500, D. 1000. Correct option is C. 500. 96. A DNA probe contains radioactive A. S35, B. N15, C. P32, D. O18. Correct option is C. P32. 97. One can find out whether human cells have a particular gene known to occur in reptiles by A. Shotgun experiment, B. DNA probe, C. PCR technique, D. staggered cut method. Correct option is B. DNA probe. 98. A cell is known to carry 6 genes that code for enzymes of the same metabolic pathway, by which method one can find out if all 6 genes are located next to one another on the same chromosome. A. By bioelastic method, B. By cloning, C. By DNA probe, D. By shotgun experiment. Correct option is C. By DNA probe. 99. The first human hormone produced by recombinant DNA technology was A. Thyroxine, B. Estrogen, C. Testosterone, D. Insulin. The correct option is D. Insulin. 100. Genetically engineered human growth hormone is now produced in large quantities. The previous source of this hormone for treating pituitary dwarfs was A. Human cadavers, B. Pig cadavers, C. Goat cadavers, D. Mutant mice. The correct option is A. Human cadavers. 101. In gene therapy, DNA is inserted into a cell to compensate for A. the lack of copy DNA, B. mutant alleles, C. the absence of plasmid, D. holes in the DNA made by viruses. The correct option is B. mutant alleles. 101. Strains of bacteria now used in gene splicing experiment cannot invade natural environment because they are A. clone, B. sensitive forms, C. mutant forms, D. hybrid forms. So the correct option for this MCQ is C.